Hello Biology 230 at Tuskegee University. This is Dr. Griffin again with the second of 3 videos for Module 12. Again, Module 12 deals primarily with transcription and translation, and in this module, in this video, we're going to be focusing on transcription. The main player of transcription is to get the message out. What exactly is that message that DNA has the potential to release for? Well, once transcribed, the primary transcript is anti-parallel to the DNA template strand. Remember, in transcription, only one of the strands will serve as a template to make mRNA. The primary transcript will eventually, in most cases, give rise to messenger or mRNA. This mRNA holds the message. Now let's get on to the point of what is the message. Well, technically, biologically, the message is a series of codons. Codons are nucleotide triplets. As you can see in your textbook, it's a nice table that gives you all the major codons and the amino acids that they will give rise to once translation has occurred. There are 3 major stages of transcription. The first stage of transcription is known as initiation. Before we break down the steps of initiation, I want to point out some major players. The first is the promoter on the template strand. This is the promoter region here, contains the start point. This is where transcription will start. And you can typically tell a promoter because it is known to have what we call a Tata box, meaning that it has lots of thymines, it has lots of adenines in it. So you should be able to readily recognize where the promoter is on your DNA strands. Now, to initiate transcription, what first has to happen is that transcription factors, and these are just proteins, these transcription factors shown here in these bubbles will directly bind to the promoter. This is mainly focusing on eukaryotic cells. Once the transcription factors have bound to the promoter region of the DNA, this allows for the recruitment and the binding of RNA polymerase II. Once RNA polymerase II binds, then the RNA transcript is free to be created. And so RNA polymerase will begin to add RNA nucleotides one by one, base by base, to create that primary transcript from the template strand only. And the whole complex containing the RNA polymerase II and the transcription factors is known as the transcription initiation complex. The second major stage of transcription is elongation, and this simply just tells the fact that the strand, the mRNA strand that's being created, shown here in red, is being elongated. That is just adding these base pairs one by one by one that are complementary to the template strand of DNA. And just one important reminder is that an RNA contains uracil and not thymine. So wherever there is a in our DNA template strand, in our new RNA strand, our new primary transcript, we will have U's base pairing with the A's and not T's. The last step of transcription is termination. So that just simply means it's ending the story. It's ending the signaling of this transcript, this message. Prokaryotes contain a specific sequence that causes polymerase to detach from RNA. That's the RNA polymerase to detach from DNA and release the transcript. And there is no further modification needed. Okay, that's a very important point. No more modification of that primary transcript for prokaryotes. However, eukaryotes contain a poly-A tail, and this is the signal for this pre-mRNA transcript, this primary transcript, to be cut away and removed from the RNA polymerase. Okay, that was pretty much a quick overview of the steps, 3 major stages of transcription. Your book has a lot of great details, and we'll discuss more in class with this hybrid course. This is officially the end of video 2 and you are almost done. You are 66.67% done with this module, and I look forward to working with you in the next video. And don't forget to view it. It's your last one for module 12, and not only to view it but to learn from the next video. Have a great day. This is Dr. Griffin signing off.